Okay, now it's urea cycle, amino group of amino acid converted to ammonia, and ammonia is very toxic for the body and must be removed. So what is the process of removal? Ammonia combines with carbon dioxide to form urea that is not toxic to the body, so the end product is urea. Urea is a major disposal form of amino group derived from amino acids and accounts for 90% of nitrogen containing compound of urine. Free ammonia provides one nitrogen of urea and another one aspartate. Glutamate is immediate precursor of both ammonia and aspartate. Nitrogen. The carbon and oxygen of urea are derived from carbon dioxide. Urea is produced by the liver and it is transported in the blood to the kidney for excretion in the urine. What are the steps in converting ammonia to urea? Ammonia combines with carbon dioxide and 2 ATP and gives carbamyl phosphate. Carbamyl phosphate P combines with ornithine and gives to citrulline. Citrulline plus aspartate gives fumarate and arginine gets converted into urea and ornithine. Most of the nitrogenous waste comes from the breakdown of amino acid. This occurs by deamination. Deamination of amino acid results in the production of ammonia. Ammonia is an extremely toxic base and its accumulation would be fatal. Liver contains a system of carrier molecules and enzymes that converts ammonia to urea, and it is called urea cycle. Urea cycle sequence, it has different catalysts. So one is CPSI, carbamoyl phosphate synthetase. The carbamoyl phosphate synthetase 1 converts the ammonia produced by glutamate dehydrogenase into carbamoyl phosphate. The formation of carbamoyl phosphate requires two molecules of ATP and take place in the matrix of mitochondria. The ammonium may come from glutamate or in free form from blood, and HCO3 comes from respiration. NH3 directly takes part in this reaction, and this reaction is called ammonia fixation reaction. You can go through the reaction, and this is the reaction given where ammonia reacts with HCO3 and gives carbamoyl phosphate. The carbamoyl phosphate is the next transfer to ornithine to generate citrulline ornithine transcarbamylase. Catalyze the condensation of ornithine with carbamoyl phosphate producing citrulline. The energy of the reaction is provided by high energy aldehyde of carbamoyl phosphate. The citrulline is transported to cytosol where remaining reaction takes place and citrulline leaves the mitochondria. Once in cytosol, citrulline condenses with aspartate. An enzyme of remaining three steps are located cytoplasm of cell. Citrulline formed in the mitochondria is transferred across the mitochondrial membrane. Now, Argino succinate synthetase, Argino succinate ligase and Arginase comes into picture. Argino succinate synthetase. The citrulline and aspartate are condensed to form Argino succinate. This reaction involves the addition of AMP to amido carbonyl of citrulline forming of activated intermediate on enzyme surface and subsequent addition of aspartate to form Argino succinate. Citrulline plus aspartate plus ATP gives back Arginase succinate plus AMP plus PPI. Organic pyrophosphate PPI contains two phosphate groups. It is unstable and it is catalyzed by inorganic pyrophosphate. This means breakdown of two phosphate groups releases energy for the reaction. Argino succinate ligase. Arginine and fumarate are produced from Argino succinate by cytosolic enzyme hydrolyzed by Argino succinate ligase. It reversibly catalyzes the cleavage of Argino succinate to fumarate and arginine. So here Argino succinate gets converted into arginine and this also supplies arginine for protein synthesis. The fumarate is reconverted to aspartate for the use in arginosuccinate synthetase reaction. Arginase. The enzyme arginase cleaves arginase from diet and from protein breakdown, generating urea and ornithine. The enzyme arginase cleaves the urea from arginine and regenerating cytosolic ornithine, which can be transported to mitochondria matrix for another round of urea synthesis. So here arginine gets mixed with water and gives us urea plus ornithine. So urea passes via transport protein into the blood and is carried to the kidney where it enters the glomerular filtrate from which it is excreted in the urine. So now the catabolism of 20 amino acids involves the removal of amino group. The next is the breakdown of remaining product that is transferred as C skeletons. The catabolism of C skeleton results in the formation of 7 different products. These are oxaloacetate, alpha ketoglutarate, pyruvate, fumarate, acetyl coenzyme, acetoacetyl coenzyme, and succinyl coenzyme. So what are ketogenic and glucogenic amino acids? Ketogenic amino acids are those whose catabolism yield either acetoacetate or acetyl coenzyme. And acetoacetyl coenzyme are ketogenic. So acetoacetate is referred as ketone body. These ketogenic amino acids are degraded to intermediates that can be utilized in the formation of ketone bodies. These products are called acetyl coenzyme and acetoacetyl coenzyme. Glucogenic, the amino acid whose catabolism results in generating pyruvate or citric acid cycle intermediate are called glucogenic or glycogenic. These intermediates act as a substrate for the gluconeogenesis and therefore give rise to net formation of glycogen in liver and muscle. The glucogenic amino acids are degraded to intermediates that can be utilized in glucose formation. These products are pyruvate, oxaloacetate, and succinyl coenzyme, alpha ketoglutarate, and fumarate. Oxaloacetate, alpha ketoglutarate, fumarate, and succinyl coenzyme are intermediates of citric acid cycle. Hence, they can be oxidized to carbon dioxide and water, and if the glucose level is low, it undergoes gluconeogenesis and form glucose. The pyruvate undergo reversal of glycolysis and form glucose or get converted to acetyl coenzyme that further oxidize to carbon dioxide and water in citric acid cycle. So nutritional state of the individual will determine the metabolic pathway taken by these products. Acetoacetyl coenzyme is oxidized to carbon dioxide and water. So it is an effort to help all Mscdfsm students for studies and other queries related to this subject. If you like the subject, please comment a thumbs up for motivation. Like and subscribe and share the channel so that more and more students get help in this corona season. For any queries, please email at kavitakundra@gmail.com. For diet related queries, patients can also email us, and for coaching, please share your phone number, email address, and in subject write down you are the coaching inquiry in mail. So all the best for your future dietitians. Take care, stay safe, stay protected. Bye bye.